There we go. So we'll be doing chapter 8. And like I said, in the lab, there is no lab today. You should be working in your unknown notes and your infectious disease paper. The infectious disease paper is due at 11:59 p.m. this Saturday, February 19th. All right, so we had talked about DNA and mentioned what we'll be discussing. Let's move in. Where did that move? There we go. So genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes, I don't think I can run this, and how genes are replicated. The gene is a segment of DNA that encodes a functional product, usually a protein, but a gene could code only for RNA. Any questions about any of that? Let me copy this to my screen, and then you'll be able to hear me better. I'm surprised nobody asked. Last time I forgot even to use it. Why don't I have that? Let me try and get it again. Oh, that's the wrong one. Now I can get rid of this. I don't know, are you seeing what I'm doing? Oops, can't do that.
Yeah, we can.
Okay. I'll close this down. Now we can get rid of it. I need to get rid of that to have a port for my headphones. I got set up to, so, right. All right, there we go. Can you hear me now?
Yes.
Okay, thank you. A genome is all the genetic material in a cell. And because we're studying microorganisms, the genome is also all the material within the organism. The study of genomes is called genomics. The genotypes are the genes of an organism, so the genes for a particular phenotype. The phenotype is the expression of those genes. Let me give an example. The genotype for blue eyes, shown here, would be the genotype is genes or alleles, lowercase b. Those code for the phenotype blue eyes. And then for brown eyes, meaning the phenotype brown eyes, shown here, the genotype is either capital B, lowercase b, or capital B, capital B. Everyone understand that? Here we're looking at the genes on a chromosome, and there's a chromosome from E. coli. The genes are on the DNA, and as you know, DNA is a polymer of nucleotides. DNA has 4 nucleotides: adenine, thymine, cytosine, or guanine, or you may call them A, T, C, and G. I don't care how you know them, but you do need to know them one way or the other. DNA is a double helix, meaning there's 2 strands of DNA, and one strand runs this way, the other strand runs this way. They're connected, each strand of DNA is connected by a sugar phosphate backbone, meaning this sugar here connects to the phosphate, which connects to the sugar, etc. That's what's holding the single strand of DNA together, and it's covalent bonds of the sugar phosphate backbone. The two strands are held together only by hydrogen bonds at the nucleotide bases. And A binds with T and forms two hydrogen bonds. C binds with G and forms three hydrogen bonds. The strands run anti-parallel, meaning one strand runs this way, the other strand runs that way. Any questions about any of that? Here's a question for you. If hydrogen bonds are holding the double strands together, and as you know, hydrogen bonds break all the time, like this A could hydrogen bond to water, and there's a lot of water in the cell. If this bond is breaking and reforming all the time, why are the two strands held together? Nobody's going to take a guess?
Is it the disfluid bonds?
It's because there's many hydrogen bonds holding the two strands together. And so when this one breaks, the other bonds are holding the DNA together, and then this one will reform, and then that one will break, or that one will break. And actually the breaking of DNA, you don't need to really know this, but they say it breathes because it'll open up and then reclose. Like I said, you don't really need to know that, but I'm just telling you. All right, the flow of different DNA in the cell, within the cell, happens that the DNA stores the information and encodes for the information. That information is then in transcription, translated into RNA, and then messenger RNA is in translation. That information is encoded into protein. So the information flows from DNA to RNA to protein. Each of these three have their own language, how the information is stored. This is known as the central dogma in biology. And we'll talk a little bit more about how DNA flows within a cell, between cells, and between generations of cells. Any question about that? All right, this slide is showing you all the ways that the flow of genetic information happens from one cell to another cell. We mentioned already that within a cell, let me blow this up, the information flows from DNA to messenger RNA to protein. Now you should realize that sometimes the information flows from DNA to rRNA or tRNA, in which case it doesn't flow further. And that's the way information flows within a cell. Between generations of cells, a parent cell will copy its DNA. The two copies of DNA will then divide, and then one will go to one side of the cell and the other to the other side of the cell. And then the cell will split down the middle so that each of the daughter cells gains a copy of the information in the parent cell. And then for humans, how we do this is two parents get together and they create the offspring. And that's how the information in DNA flows from the parents to the offspring. Any question about any of that? Now, the DNA can also flow between cells of the same generation of cells. And I've changed the picture from the textbook to make it a little clearer. There is a donor cell where the DNA comes out of that donor cell and then recombines in a recombinant cell. So this is the recipient cell. And what I've done is I've broken this because the DNA doesn't flow directly from this cell into that cell, at least not most of the time. Most of the time, the DNA flows out of this cell, and then we have naked DNA, and that naked DNA can come into a recipient cell, and this we call the recombinant cell, and then this cell can gain new DNA from another cell. Everybody got that? And this is when DNA flows between cells of the same generation. We'll talk about each of these in a little more detail. So the flow of information within a cell happens from DNA to RNA to protein. And the language of DNA is nucleotides. There are 4 letters or nucleotides in the alphabet of DNA. The information flows to RNA. The language is still nucleotides, but it's RNA nucleotides instead of DNA nucleotides. And there's still 4 letters in this alphabet. The information then flows from RNA to protein, and the language is amino acids. And now there are 20 letters in the amino acids. And for comparison, the language of English has 26 letters in the alphabet. Any question about any of that? So for DNA, you could say that is a dog. In RNA, it would be similar, but with a different dialect. That is a dog. And then the translation from RNA to protein, it would be translated to something like quello è un cane. The same information, but in a different language. Any question about any of that? All right, so we talked about the flow of information within a cell. Now let's talk about the flow of information between generations of cells. The DNA stores the information, and that information is passed to daughter cells as the DNA replicates, and then the cell divides. Let me blow this up. How the DNA replicates is a DNA polymerase comes onto the DNA and will make the DNA single-stranded, so the 2 strands separate. We'll talk a little bit about that in a minute. But the 2 strands separate, and then the DNA polymerase looks at the nucleotide base on the template strand of DNA, meaning the old strand of DNA, and then adds the corresponding nucleotide to the new strand of DNA. So if G is on the template, the DNA polymerase will add C to the new strand of DNA. And then when it moves down the nucleotides and sees a C here, it'll add G. And then it'll move down and see an A here, then it'll add T. The DNA polymerase, when it adds these nucleotides to the new strand, they are added as a covalent link between the sugars and the phosphates. The DNA replication is called semi-conservative because when the DNA replicates, one strand is the old strand, and it's used as a template to make the new strand of DNA. So the dark strands are the old strand of DNA. And then when the DNA replicates, the white strand of DNA, the daughter cell will get one white strand or a new strand and one old strand or the dark strand. Any question about that? So DNA replication is semi-conservative. Conservative would be replication. That model would have been where one strand or one of the newly replicated DNA, both of them would be light strands and both would be dark strands. So that would be the conservative. And don't ask me what the liberal model is. I have no idea. So when the nucleotide is added, it's added as a triphosphate, and the DNA polymerase adds the correct nucleotide to the template strand, and then it links this phosphate to this sugar group here. And the DNA is put down on the new strand in the opposite, what do you call that? Opposite way as the old strand, meaning this strand is going this way, this strand is going that way. And then the double strand is not covalently linked. It's only linked at the nucleotides by hydrogen bonds. Let's see, is that everything? All right, let's watch a little movie on DNA replication. There are 3 enzymes you need to know. Helicase, the polymerase. Boy, that doesn't look like it's good. Three enzymes you need to know: helicase, the polymerase, and then ligase. The movie will probably talk a little bit more.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any question about that movie? I'll have to try that again. For some reason, when I click on the link, it doesn't seem to work, but I can take the information on the link and then get it to work. So the movie discussed a few other enzymes. You only need to know 3 of the enzymes. Helicase, which separates the DNA and makes it single-stranded. DNA polymerase, which makes the new strand of DNA by reading the template strand of DNA, and then DNA ligase. DNA ligase, what it does is it links a break in the DNA together. Where the first DNA comes down, let's say we separate the DNA here and make it single-stranded. And which way is this one going? It looks like it's going that way. So this DNA comes here. That means the DNA polymerase comes here. It'll start a new strand of DNA moving this way. And there will be a break from the DNA of the old strand of this one here. And then on the, that's the leading strand. And then when this DNA stops, there will be a break in the DNA. So in the leading strand, there are not too many breaks in the DNA, but on the lagging strand, there's a break between each Okazaki fragment. So this would be a break, and that would be a break, and then that would be a break. Wherever there's a break in the DNA, the ligase comes in and ligates or seals the DNA together. And that's a very strong linkage because that's the covalent linkage between the sugar and the phosphate backbone of the single strand of DNA. Any question about any of that? The DNA is only made in one direction. In one strand of DNA, the DNA polymerase can make the DNA sequentially. So the DNA, binds to the single-stranded DNA and then moves down it this way, adding on the correct nucleotides for the newly replicated strand of DNA. On the other strand, called the lagging strand, the DNA polymerase moves this way, and the DNA is separating that way, so this isn't going to work. And what the DNA polymerase does is it makes the DNA in Okazaki fragments, and so the DNA will bind here and then go to here, and a DNA polymerase will bind here and then go to here. And so the lagging strand is made in short patches all the way down. Any question about any of that? Let's see. I think that's about it. The new DNA is made in the 5' to 3' direction. It is initiated by an RNA primer, but I'm unlikely to ask you about any of that. And there's a leading strand where the DNA is synthesized continuously, and a lagging strand where it is synthesized in short patches or discontinuously, resulting in Okazaki fragments. And then the DNA is joined together by DNA ligase. Wherever there's a break in the DNA, ligase will seal that break. All right, any questions about DNA replication? Can everybody hear me?
Yeah.
Okay, thank you. So transcription is the next topic we'll talk about, and that is where the information is copied from DNA to make RNA. DNA is transcribed to make RNA, and there are 3 types of RNA made: messenger RNA, tRNA, and rRNA. Here's a question for you. When the RNA polymerase binds to the DNA of a gene, how does that DNA polymerase know whether to make messenger RNA, tRNA, or rRNA? Does anyone know? Well, what's the difference between messenger RNA, tRNA, and rRNA? Is there any difference?
Don't they have different jobs?
They have different jobs, yes. Do they have the same RNA nucleotides?
No.
So you're saying this one will have guanine, but that one will not?
Are they different structures?
They are different sequence. Messenger RNA has a different nucleotide sequence than tRNA, but they are the same RNA nucleotides.
Oh.
Okay, so they are the same RNA nucleotides, but they do have a different sequence. And how the DNA, or I should say the RNA polymerase, knows which RNA to make is it just reads the gene. If it's a gene that codes for protein, the messenger RNA will be made. But if it's a gene that codes for tRNA, tRNA will be made. And if the gene codes for rRNA, rRNA will be made. So it's very easy for the RNA polymerase. It just reads what the gene is. And that's the RNA it makes. And there's no difference between these 3 RNAs other than their nucleotide sequence, meaning they'll have a different sequence of nucleotides. And yes, they do have a different function, and that's related to the sequence of the nucleotides. Transcription begins when the RNA polymerase binds to the promoter. A promoter is a region upstream of the information that we call the gene. The RNA polymerase binds to the promoter, then moves down the DNA, transcribing the DNA to make the RNA. Transcription proceeds in the 5' to 3' direction, the same direction as DNA replication. So RNA is made from 5' to 3'. And the RNA polymerase will move down the DNA, transcribing it, making RNA, until the RNA polymerase reaches the terminator. The terminator is a region of DNA after the end of the gene, and the terminator codes for the RNA polymerase to fall off the DNA. And when that happens, obviously transcription stops, and the RNA is now free from the DNA and the RNA polymerase, and then it can move to where it wants to move. Any question about any of that? Only the genes are transcribed into RNA. So not all of the DNA is transcribed into RNA. Only the genes are. Non-coding DNA is not transcribed. And in eukaryotes, most of the DNA is non-coding DNA. In prokaryotes, there's actually little, it's a minor part of the DNA, non-coding DNA. But there is some non-coding DNA. And that non-coding DNA will not be transcribed. Any question about any of that? In DNA replication, all the DNA is replicated. But in transcription, only the genes are transcribed. Here we're looking at a picture of transcription, DNA being copied into messenger RNA. And there we see it in an actual electron microscope. It's using fluorescent dyes to show this. Any question about any of that? All right, so let's look at transcription in a little more detail. See if I can grab this here and see if this will work, because if I open it directly, it doesn't seem to work. Transcription. That doesn't look promising. Yeah. Did I spell that right? I think this is the one I want.
Replace your notebooks and printed documents with Remarkable 2, the only tablet that feels like paper.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns.
Okay, we'll talk about exons and introns in a little bit. Let's not get into it now. All right, any questions about that movie? It was good in the sense that it discussed promoters and terminators and then showed you where they are on the, well, in relationship to the gene. All right, so RNA transcription begins when the RNA polymerase binds to the terminator. And the terminator is a place that codes for the polymerase to bind to the DNA. That RNA polymerase then moves down the DNA, and I guess that would be the DNA here. And it uses the DNA as a template to make the RNA strand. So it puts on the complementary base of RNA. And then as it moves down the strand, it adds more nucleotides. And that RNA polymerase will move down the gene until it reaches the terminator. The terminator codes for the RNA polymerase to fall off the DNA. When that happens, the RNA is released from the DNA, and of course the DNA snaps back together as a double helix. Any question about transcription? Okay, now the messenger RNA then moves into the cytoplasm, at least of a eukaryotic cell. It's already in the cytoplasm of a prokaryotic cell. And then we can have translation of the messenger RNA occurring. Now remember, transcription doesn't always make messenger RNA, only if this gene codes for a protein. Transcription can make tRNA or rRNA if the gene codes for tRNA or rRNA. All of the messenger RNAs, meaning tRNA, rRNA, and messenger RNA, get together to perform translation. Translation is the synthesis of proteins by ribosomes using the information encoded by the messenger RNA. Messenger RNA is read in groups of 3 nucleotides that we call a codon. So one messenger RNA nucleotide is not read, they're read as groups of 3, and we call this a codon. Translation of messenger RNA begins when the ribosome moves down the messenger RNA. And then translation will begin when we reach the start codon. The start codon is AUG, 3 nucleotides in the messenger RNA that tell the ribosome, start protein translation here. So what happens to the messenger RNA before AUG? What does the ribosome do with it? Why did I, there we go. Nobody has a guess? So when the, let me blow this up a little. When the ribosome binds to the messenger RNA and then moves down it, all of the first messenger RNA is ignored. It's not read. Protein translation does not begin until the ribosome reaches the start codon, which is AUG. And you should probably know this. The start codon is AUG. AUG does more than tell the ribosome to start protein translation here. AUG also encodes for the amino acid methionine. So AUG has 2 functions. And so every protein that gets made, they all start with the amino acid methionine. And if that protein doesn't want to start with the amino acid methionine, the cell will have to remove the methionine from that protein. But all proteins, when they're made, they always start with the amino acid methionine because AUG codes for methionine. The ribosome will move down the RNA and it will translate the messenger RNA from AUG all the way to the stop codon. When the ribosome reaches one of the stop codons, and there's 3 of them, you don't need to know what they are, but there's a stop codon like UAA, will tell the ribosome, stop protein translation here. And then protein translation stops. When protein translation stops, meaning when it reaches the stop codon, the ribosome will disassociate. That will release the protein from the messenger RNA, and then the protein will go and do whatever it's supposed to do, or move to wherever it's supposed to move in the cell. Any questions about any of that? All right. So as I mentioned, the messenger RNA is read in groups of 3 nucleotides that we call a codon. Each messenger RNA codon codes for 1 amino acid. But there's a problem, and that is there's only about 20 different amino acids, but there are slightly over 60 codons. What does the cell do with all the extra codons? Well, what happens is there's more than 1 codon that codes for most of the amino acids. For example, UUU codes for the amino acid phenylalanine, but UUC also codes for that same amino acid. There are 2 amino acids that have only 1 codon. Methionine only is coded by AUG, and tryptophan is only coded by UGG. You don't need to know that. Some of them, like phenylalanine, have 2 codons that code for the amino acid. Some of them have 4 codons, like serine. And then leucine, there are 6 codons that code for that amino acid. So some of the amino acids, actually most of the amino acids, have more than 1 codon that codes for that amino acid. This is known as the degeneracy of the code. Any question about any of this? So let's talk a little bit about protein translation. I have a little video to show you. It's a bit loud.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about the video? If not, let's go through it. Let me shrink that. So translation occurs as the ribosome and there's 2 units formed together on the messenger RNA. And then that ribosome moves down the messenger RNA until it reaches the AUG site. Another component that's important is the tRNA. The tRNA is important because it's actually the thing that reads the messenger RNA, meaning the tRNA anticodon binds to the codon, and that's only a hydrogen bonding link, but the ribosome stabilizes the link. And the tRNA brings in the correct amino acid corresponding to the codon. Any questions about any of that? The ribosome has 2 openings in it, and the first tRNA comes in the first opening and binds to UAG, meaning the first tRNA will have the anticodon UAC, which binds to the codon AUG. And the first tRNA brings in the first amino acid, which is always methionine. It is the ribosome that moves down the messenger RNA until the AUG. And then the first spot in the ribosome opens up over the AUG so the tRNA can come in and bind. The second tRNA then comes in the second spot of the ribosome, and it'll be the correct tRNA that'll bind to the second codon, and this codon can vary for what it is. When that tRNA brings in this second amino acid, the ribosome then removes the methionine or the amino acid from the earlier tRNA and then covalently links it with a peptide bond to the amino acid on the second tRNA. So the methionine is linked to the leucine amino acid, the second amino acid. This is something we discussed earlier, and that is the enzyme that does this activity is actually the RNA portion of the ribosome. So this is the only enzyme we will discuss that is an RNA enzyme. And your textbook doesn't discuss that, but I'm just mentioning it to you. This is the only time we'll ever discuss an rRNA enzyme. All right, let's move on. So when that happens, the first tRNA that no longer has an amino acid linked to it will then fall out of the ribosome or move out of the ribosome, whatever you want to call it. And then the ribosome will move down 1 position. And the third tRNA will move in. So now what was the tRNA in the second position is now in the first position of the ribosome. And then the third tRNA can come into the second position of the ribosome, and it'll have its anticodon bind to the codon. And then the ribosome will remove the leucine from the tRNA, or I should say the second amino acid from the tRNA, and then covalently link it with a peptide bond to the amino acid over here, on the third tRNA. This will allow this tRNA to move out. Is that shown there? Nope, shown here. And then the ribosome will move down one position so that the next codon is in the second position of the ribosome. And then that tRNA will come in and we'll have a growing polypeptide chain. This process will continue as the ribosome moves down the messenger RNA by one codon, moving the second position to the first position. And of course, the tRNA of the first position comes out. And then that'll allow another tRNA to come in and bring in the correct amino acid. This will continue until the ribosome reaches the stop codon. Once the ribosome reaches the stop codon, it will release the protein from the, the tRNA and the messenger RNA, and then the ribosome will disassociate and fall off of the messenger RNA, shown here. Protein is separate and the ribosome falls apart and the last tRNA comes out. Any questions about protein translation?
Sorry, I missed. What was the fourth step?
I'm not sure. What step are you referring to?
Oh, I was going off the numbers right there. I thought those were the steps.
This is actually the seventh step.
Okay.
Okay. All right. If there's no questions, let me talk a little bit about a complication. As you know, DNA is transcribed into RNA, and then the messenger RNA is translated into protein. Well, here we have a gene, and as you can see, it is being transcribed into 1, 2, 3, 4, 5, 6, 7, 8 different messenger RNAs. So that's a complication. And that is one gene can have more than one messenger RNA. I'm talking about a gene coding for a protein. And then in prokaryotes, it's even more complicated because this process of transcription is occurring in the cytoplasm of the cell. And here you can see a prokaryote where here's the DNA and the messenger RNA is being made. And here we're seeing 1, 2, 3, 4 different messenger RNAs being made from the same gene. And this is the larger messenger RNA because the polymerase has had more time to make the messenger RNA. This polymerase is just getting started making the messenger RNA. But in prokaryotes, you can notice that ribosomes can bind to the messenger RNA while it's still being made. And then the ribosomes are in the cytoplasm, so they can start translating the messenger RNA even before the messenger RNA is completely transcribed. And so you have in prokaryotes protein translation occurring while RNA transcription is going on, meaning while it's occurring. We have both protein translation and messenger RNA transcription occurring at the same time. That's a complication. Now in eukaryotes, transcription and translation never happen at the same time because in eukaryotes, transcription is happening in the nucleus and translation is happening in the cytoplasm. Any question about any of that? In eukaryotes, we have another complication though, and that is in a gene, we have regions of the gene which code for the messenger RNA and code for the protein, and we have regions of a gene that do not code for the protein. This region is called an intron. The region of a gene that codes for the protein is called an exon. When the RNA is transcribed, both the exons and the introns are transcribed. And before this RNA can leave the nucleus, the introns have to be removed. We call it that the introns are spliced out. That means they're cut out. And then the exons are spliced together or they're put together so that the final messenger RNA of a eukaryotic cell only includes the exons and only the final messenger RNA is allowed out of the nucleus. The pores in the nuclear membrane will not allow an RNA to leave the nucleus if there are introns in that RNA. Any question about any of that? All eukaryotes have introns in their genes. Bacteria tend not to have introns, so we're going to just say bacteria do not have introns in their genes. Any question about any of that? All right, we talked about the structure and function of the genetic material. Let's move on to the regulation of bacterial gene expression. That happens with an operon. So there are different ways a cell can regulate what it has within it. And an easy way to regulate what's in it is to regulate the expression of the bacterial genes. So regulate a cell by controlling gene expression. There are some genes that are always turned on and are expressed at a fixed rate. We call these constitutive genes, like an enzyme would be a constitutive enzyme. It's always made. The gene is always turned on. There are other enzymes that are expressed only as needed. They are turned on when the cell wants to make them, and they are turned off when the cell does not want to make them. These are controlled by repressible and inducible genes. So an enzyme can be a repressible enzyme or an inducible enzyme. There are other ways that the cell can control the expression of genes. You can regulate by pre-transcriptional control. Oh, excuse me, that's what I've already talked about. Inducible operons, which we'll talk about. You can regulate the genes by a repressible operon. We're not going to cover that. You're not going to be tested on it. You can also control a gene by epigenetic control. We're not going to discuss that, and you're not going to be tested on it. And then there's post-transcriptional control. We're not really going to test you on that. We will briefly discuss it, and that's where a gene can be made, you make the protein to it, but then you either shut down the protein or you degrade the protein. So not all genes are turned on all the time, and it doesn't make sense for a cell to turn on a gene product if the gene product is not going to be needed. And so the cell will turn off and on specific genes. How the cell does that is with a bacterial operon. An operon can be thought of as a supergene. It does contain the structural gene of interest. And normally when we talk about a gene, we're talking about a structural gene. A structural gene is specifically what we're talking about. Like in this case, Z codes for the enzyme lactase. If we're talking about the blue-eyed human gene, we're talking about a structural gene. Any questions there? So an operon does contain one or more structural genes. We'll discuss about prokaryotes and eukaryotes in a little bit. The operon also contains controlling regions of DNA that control the expression of the structural gene or genes. So the lac operon contains actually 3 structural genes, gene Z, Y, and A, and they're all involved in the metabolism of the sugar lactose. And that's always seen in prokaryotes, that if you have more than one structural gene in the operon, these genes work together. And then the operon contains the controlling regions, such as the promoter, which we've already discussed, and the operator. These regions control the expression of the structural gene. And then not shown here, the terminator is part of the operon, and it codes for the RNA polymerase to fall off the DNA. Okay, any questions about any of that? If not, let me state that the lac operon, which we're talking about, is an inducible operon. The gene is normally turned off, but it can be induced to be turned on. The lac operon contains 3 structural genes. The first, Z, codes for the enzyme lactase, which can break the sugar lactose into the sugar, beta galactose and glucose. The gene Y, the structural gene Y, is the permease gene, which codes for the transport protein that brings the sugar lactose across the cell membrane. So the Y gene codes for the protein which transports the sugar into the cell. The A gene helps in converting beta galactose into the sugar glucose. So all 3 of the structural genes are involved in the metabolism of the sugar lactose. The promoter and the operator control the expression of these genes, and we'll talk about that next time. And you'll note that in the lac operon, we also have a region near it that's called the regulatory gene. The regulatory gene is not a part of the lac operon. It is its own operon, and it makes a regulatory protein that regulates the expression of the lac operon. And we'll talk more about the regulatory gene later, next time. For now, let me mention that normally the regulatory gene that controls an operon is not near the operon. In fact, in eukaryotes, the regulatory gene is usually on a different chromosome than the operon is on. But in the case of the lac operon, the regulatory gene is right next door to the lac operon. All right, any question about any of that? If not, I'm going to stop here and we will start from here next time.
I have
Okay.
a question, but it doesn't have to do with this.
Okay,
Can you clarify for me how many ATP are created by the Krebs cycle?
can you repeat that question? I didn't quite hear all of it.
How many ATP are produced by the Krebs cycle?
Oh, okay. 2 ATP are made by the Krebs cycle per molecule of glucose.
Okay, per molecule of glucose.
You got to remember that glucose is broken down into 2 pyruvates. So 1 pyruvate only makes 1 ATP, but there are 2 pyruvates that are made from glucose.
Okay,
Okay,
thank you.
all right. Anything else? If not, I'll see you next time.
Have a good night.
You too.